Okay, for Mendel's work, we have some principles, or kind of things he found out. Genes are responsible for all inheritable characteristics, so genes are responsible for your traits. Phenotype is based on genotype. In other words, your genotypes make the way you look, or your genes make the way you look. Each trait is based on 2 genes, one from the mom, one from the dad. Like I said before, you get 1 chromosome from mom, you get 1 chromosome from your dad. On your chromosomes are your genes. So for every trait, you have 1 from mom, you have 1 from dad, which is why we use the 2 letters when we're making the genotypes. A true breeding individuals are homozygous for both alleles. They're homozygous. They have the same allele. Now remember, allele is just one of the forms of the gene. Blue eye, or for eyes, you have blue, you have brown, and you have green. So there's 3 possibilities, or 3 alleles. Of that, you would only get 2, 1 from mom and 1 from dad. But true breeding individuals, they have the same. And remember, with Mendel, he took that true breeding plant and crossed it with another true breeding plant. So like pure tall with pure short, pure round with pure wrinkled, pure yellow with pure green. Big letter crossed with little letter. That was P1. His P1 cross, or making those have babies, gave him the F1 kids. So when you took big T crossed with little t, and you do the Punnett square, and the Punnett square are all possibilities of their children, those children are known as F1. He then would take F1 and cross it with another F1. That was his F1 cross, which gave you the F2 babies. The law of independent assortment, it was talked about earlier in yesterday's video. It's the genes for different traits are inherited independently of each other. So the genes that you got for your eye color did not influence the genes that you got for your hair color, which did not influence the genes you got for your skin color, which did not influence the genes for how tall you are. The law of segregation. During the formation of gametes, or remember your eggs or your sperm, the 2 alleles responsible for a trait separate away from each other. The next slide is going to show exactly what I mean by that. Allele for a trait are recombined at fertilization. So they separate out to make the egg and the sperm, and then when they have sex, the egg and the sperm come back together to make the babies. So let's look at that. Okay, so you can see here at the top you have a female parent, and off to the side you have a male parent. Tall is dominant over short, and you can see that with the female parent, she would produce eggs. One of her eggs is a big T. Her other egg has a little t. So those 2 alleles were one time together have now segregated or separated out in forming of the egg. For the male, he had a big T, little t. One of the big T went to one sperm, the little t went to a different sperm. Again, they were together in that male. When the sperm were made, they separated out away from each other or segregated. So the law of segregation is really being applied to forming of the egg or the sperm. They then come back together during fertilization. So when you have the egg meeting up with the sperm, they're now together to make a child. This actually, Punnett square, is a representation of one of Mendel's crosses, and it would be his F1 cross, because the female's big T, little t, the male's big T, little t. So inside those boxes are all possibilities of their kids. That would be the F2 generation. Incomplete dominance. Okay, now we're going to start with something completely new today. So far we've been working with Mendel and his P's. We've had 2 alleles, 1 dominant, 1 recessive. It's going to change a little bit because life is not that easy. So if you don't know already, in means not. So what we're saying here is something is not completely dominant. Not completely dominant. Incomplete dominance. I'll say that again. Incomplete dominance is when an allele is not completely dominant over another allele. You may want to write that down. Okay, so you have F1 hybrids have an appearance somewhat of in between the phenotypes of the two parental varieties. I'll give you an example in a minute. We're going to be talking about snapdragon flowers. So snapdragons show incomplete dominance. Hair shape is one for humans that show incomplete dominance. So let's say you have the flower. You could have a red flower, which is big R, and you cross it with a white flower, which would be little r. And what we've been talking about so far, you would expect their babies to be big R, and that they would look red. But in this case, the gene that makes the red color is not completely dominant over the white. It cannot completely overpower the white. So they blend together and you will have a pink flower. Incomplete dominance. For the test, you'll have to know which ones we talk about, which ones are incomplete dominant. So snapdragons, you have to know snapdragons are incomplete dominant. You'll have to know that hair shape is incomplete dominance. Those are the two main ones we talk about. There's a lot of them out there, but those are the two main ones we'll be talking about. So again, big R is red. Little r would be a white flower. But when you mix those, so if you do a P1 cross, you have the pure red with the pure white, and you mix them together to make an F1 generation, they are all going to be pink. In the box, you would have big R in all those boxes for the F1 generation. Okay, there's the Punnett square. So again, big R was crossed with little r. So you had pure red flowers. They are pure with a pure white flower, but they are going to make, for the F1 generation, this would be P1. You'd label this as P1, not because we did it first, but because you have a pure with a pure. P1 with P1, you get an F1 generation. So all of these children that could be produced, this is everything that could be produced, is the F1 generation, and they would be pink, because that big R cannot completely overpower the little r in this case. Here's some pictures of snapdragons and kind of from generation to generation. So you can see with the parental, with the P1, the first set we have, you have the pure red with the pure white, and no matter what, you will always get big R, little r, and they always turn out pink. Well, if you were to follow what Mendel did with his peas, you would take an F1 and you would cross it with an F1. So you would take a big R, little r, and you would cross it with another big R, little r. That would be known as the F1 cross. Okay? Because it's heterozygous crossed with heterozygous. If you do that and you do your Punnett square, you will see that you would get 1 out of 4 times red, or 25%, 2 out of 4 times pink, or 50%, and 1 out of 4 times white, or 25%. And that's what this is showing you down here. And then you could also take a pink flower if you wanted to, which would be big R, little r, and crossed with a white flower, which is little r, and if you did that Punnett square, you would see half the time you'd have pink flowers and half the time you would have white flowers. This is not an F1 cross. You do not have heterozygous crossed with heterozygous. You have a heterozygous crossed with a pure. This is not an example of a Mendel F1 cross. But it is something you may want to do. If you want only pink and white flowers in your garden, take 2 F1 generation kids and mate them with them, and mate it with a white flower. Okay, I'm going to talk about hair color before we move on to codominance. I'm just going to use this screen. With hair color, or not hair color, hair shape is another example of incomplete dominance. Okay, there are some questions about that. So curly hair would be like big R. Straight hair is big R, little r. But that big R gene, the gene that actually codes for curly hair, cannot completely overpower the one that codes for straight hair. So you will have wavy hair. Okay, another incidence that's a little different than just pure dominant versus pure recessive, like with Mendel P's, is something that's called codominance. And in this case, it means both. So both are dominant. Both genes are dominant, so they both have to show up. 2 alleles affect the phenotype in separate and distinguishable ways. Neither allele can mask the other, and both are expressed in the offspring. There's these erminate, they're called erminate chickens. They have black flowers, black feathers. They could have white feathers. But when you get the heterozygous, since the black and white are both dominant, they will have both black and white feathers. Erminate chickens. I can show you an example at a later time. But they are chickens that have both black feathers and white feathers. Codominance. You know some animals, they have multicolored fur. Some of it's due to codominance. Both or all the colors have to show up. So 2 alleles are expressed in a heterozygous individual. Blood type is another example. Kind of ignore the I's in this case and just look at the letter A, the letter B. Okay, so when it comes to people who are AB, they have blood type AB, that's codominance. A and B are codominant to each other. They both show up. That protein is on the red blood cells, and both of them show up. But when you talk about A, someone can be AO, and only A shows up, because A is completely dominant over O. B is completely dominant over O. But A and B, when they're together, that's codominance. They are equal. And this is just showing you a cross of for blood. In cattle, roan cattle, that's mixed red and white hairs. So red is big R, white is little r. They have babies, you're going to have big R, little r babies that have both the red and the white coat. It's a similar example as with the chickens. And you can read this question and you can answer it, and then I will let you know if you're right or wrong. Okay, that's where I want to stop.